Hello, everyone. Welcome to another exciting edition of our monthly Microbial Insights webinar series. I'm your host, Dexter Puckett, project leader at Microbial Insights. And today we are thrilled to have with us a brilliant and experienced remediation expert and scientist, Dr. Michaeline Albright. To build the mystery and suspense, here is today's presentation title, Allonnia's Nature Detectives at Work, Finding New Microbial Solutions for Oil Sand Processes and Water. We are very excited to be working with Allonnia to develop specialty qPCR assays to help document and monitor their proprietary biotech solutions. MI thoroughly enjoys working with companies like Allonnia to develop tests which can be used to better understand and document microbial processes. And we have worked with Kent Sorensen and their leadership team for over 20 years. So it is truly an honor to have their company present on our webinar series today. Dr. Michaeline is a research and development team lead at Allonnia. She is passionate about using microbes as solutions to global challenges. Michaeline's research background is in microbial ecology. She holds a bachelor's in geological science from Brown University and a PhD in biological sciences from the University of California, Irvine. Prior to joining Allonnia, she was a scientist at Los Alamos National Laboratory, where her research group focused on exploring microbial community dynamics with a goal of microbiome engineering, which is improving the function of an ecosystem by manipulating microbial composition. As many of you have attended before know, this webinar today will be recorded and available on the resource section of the Microbial Insights website within the next week. A link will also be emailed to all registrants as soon as it is available. At the end of this morning's webinar, there will be a question and answer period. Please type your questions into the GoToWebinar software and our speaker will answer them at the end of the presentation. You can also email any additional questions to info@microbe.com. All right.
Great. Well, hello, everyone. Thank you for being here. I'm really excited to have the opportunity to present some work that our team at Allonnia has been doing on the bioremediation of oil sands processed water. To get started, I'm going to first introduce you to Allonnia. So we are a bioingenuity company that's dedicated to solving the world's toughest waste challenges with the world's smallest organisms, microbes. And our goal is really to take the most difficult unmet needs and deliver viable commercial solutions. We're applying our ingenuity through biotechnology and engineering with the goal of allowing industry to transform liability to value and to reduce their planetary impact. Our motto is, waste is a failure of the imagination, and we believe that we can work in harmony with nature to fast forward time and to bring nature's future solutions to the present. And so what do I really mean by that? Well, we know that microbes can live in some of the most challenging, extreme, and contaminated environments, and that they're able to evolve the ability to metabolize many different compounds. And so, however, that process is often very slow. And really, we want to take that and speed it up and make it so that these solutions, these are viable solutions that we can use in our world today. At Allonnia, we're targeting three main areas: remediation, upcycling, and sensing. And we're working in three important sectors, including mining, and here we're both upcycling critical elements and cleaning up legacy assets. In industry, where we're breaking down forever chemicals, also known as PFAS, and then plastics, where we're working on upcycling nylon and polyurethane foams. And so across all of these markets, our goal is really simple: to help bring industry and biology together to perform better. So this is ambitious. So how do we do it? We approach these challenges through combining bioinformatics, transformative biology, and integrated systems. Through bioinformatics, our goal is to discover. Through transformative biology, our goal is to develop a solution. And then through integrated systems, we're working to deploy these solutions. Today, I'm going to dive deeper into research that's happening in one of our early success stories, where we're using this discover, develop, and deploy strategy for remediation in mining. And so let's start with the problem. So oil sands process water is a byproduct of the bitumen extraction process. And through this process, more than 1.3 billion cubic meters of tailing water have been stored in these sprawling man-made ponds. So just to give you an idea of how much water this is, it would fill 520,000 Olympic swimming pools. And what's more, this water is highly toxic, so it can't be released back into the surrounding kind of environmental streams and waterways, as it would be detrimental to the wildlife, killing the birds, the rainbow trout, and also it would harm communities, people who are living downstream of these large ponds. It's thought that much of the toxicity in OSPW is due to naphthenic acids. And so naphthenic acids are a group of organic pollutants. They're comprised of saturated and unsaturated alkyl substituted acyclic cycloalpha-fattic carboxylic acids. That's a mouthful. But basically, they naturally occur in these hydrocarbon deposits, and they have widespread industrial uses. However, they're released during the bitumen extraction process when we have really large volumes of hot water that are mixed with sodium hydroxide. And so this extraction process has created an environmental challenge. And our overarching goal is to develop a low energy technology that can detoxify this OSPW by removing or transforming the NAs and any other toxic organic compounds that are found in this water. So as the Allonnia team, so generally biodegradation of NAs is considered a promising remediation technique. It is passive, effective, and environmentally friendly as a way to treat the massive amounts of OSPW water that has really accumulated. It's being produced now, but it has also just accumulated over the decades of mining. And so a biological solution avoids the use of chemicals and other materials that would need to be disposed of, whereas biology can do it without this creation of these other byproducts. Biological remediation of NAs is sustainable and cost effective, and we know that biology works. So when we have OSPW ponds where we're not adding kind of new water and new naphthenic acids from the bitumen extraction process, we see that over time, the toxicity is gradually attenuates. And so microbes are doing something, and they are helping this issue. However, it's really, really slow. And because as I mentioned at the beginning, biology isn't always fast. And so it isn't a plausible solution for these oil sands mining operations who are really under pressure to figure out what to do with this growing amount of toxic water right now. At this point, viable microbial solutions haven't been identified. Much of the previous work has started with identifying microbes that work well in commercial or synthetic NAs, and these really haven't translated. They've been a poor proxy for OSPW water. So here I've shown a figure where we have a graph of these less complex NAs, there's a microbial addition, and in about a month, we're seeing near complete degradation of the NAs. However, when these same microbes were added to complex OSPW NAs, over 3 months, these researchers only observed a 50% decrease. So this is where we started as the Allonnia team stepped up to this challenge of detoxifying OSPW. And we started in the discover phase. And as part of this phase, the first thing that we did was we sequenced a bunch of OSPW water samples to see what's there, what are we working with. We found that OSPW is home to a diverse microbial community. In this community, it includes over 50 different genera, and shown here in this bar plot, I just have the 20 most abundant taxa. And again, another thing you can see here is that there's a relatively even distribution of some of these most abundant taxa. It's not like one microbe is just dominating the whole community. So this was an exciting start. There are a lot of microbes there that we can try to work with. What was even more exciting was the diverse metabolic potential represented by these taxa. So we have sulfur reducers, we have nitrogen fixers, we have methylotrophs, ammonia oxidizers, and of particular interest to us, we have hydrocarbon degraders. So starting with this diverse community, we went about trying to enrich and isolate microbes that degrade naphthenic acids and detoxify OSPW. And we approached this by starting with OSPW NA concentrate. So we wanted to avoid learn from some of the previous research that has been done. We know that the synthetic NAs aren't a good proxy. However, this OSPW NA concentrate gives us this high concentration of naphthenic acids to add to water and allow us to try to select for microbes that, one, can survive in the presence of these NAs, and secondly, hopefully actually metabolize them and use them as a carbon source. So starting with this OSPW NA concentrate, we had a lot of different setups where we added different nutrients, micronutrients, macronutrients, beads to have biofilm growth on, kind of change the conditions in a lot of different ways, and did these transfer enrichments over time to really try to get a microbial consortia that can use the NAs and the other organic compounds in the OSPW to grow. From these consortia, we then actually went on to isolate microbes, and in these isolations, kind of a cool story here and something unique that we did was normally isolations are done on agar gels with whatever additions of either rich media, poor media that you're, depending on what you're trying to isolate. However, we actually use silica gels. And so the advantage here is that there is no other carbon source. The agar contains some carbon. The silica gels were able to remove all other carbon sources and just spread a light film of this Na concentrate or the OSPW on top of them. And then inoculate with the enrichment and get isolates from there. So we found some consortia, some isolates, and here our next steps are really to try to better understand the metabolic potential of these isolates and consortia through further sequencing. At the same time as we're doing this, we're also going to this next stage of the transformative biology, or the development. And this is where we're looking at the ability of these microbial consortia and these isolates to detoxify OSPW. Kind of our first indication that we have something that we're excited about is that we identified isolates and consortia that are growing in OSPW by itself. And here NAs are the only organic carbon sources. And so this is, I guess, NAs and other complex recalcitrant carbons are the only carbon sources. And so this is an example of some data where we have optical density on the y-axis, we have time on the x-axis, and we see that with our microbial treatment, this is an example of a isolate, and you see rapid growth over just a week compared to the OSPW control. And then shown pictured on the right is, again, you have this kind of clear OSPW control, no growth, and then you have the microbial treatment, which is turbid with all the cells growing. If you worked with OSPW, you know that OSPW water doesn't always look this clear. In this example, we started with, in these experiments, filtered OSPW to kind of remove all other, to make it a simpler system and to be able to see this growth. However, then we kind of transferred to using from these initial systems and the filtered OSPW to actually using OSPW with no filtration and instead using ATP assays and other methods to look at microbial activity. And we're still seeing similar results in these more complex systems. So we see growth, and then the question is, are they detoxifying? And here we got some more exciting results, which are that the microbes are, as they're growing, they're detoxifying the OSPW. And so this graph on the y-axis, we see the effect on bioluminescence. And here, when we have a reduced effect, that means there's a reduction in toxicity. And on the x-axis, I have just a few different samples. We have untreated OSPW, which is our starting point. And then we have several of the different isolates and consortia shown here. And we see up to a 45.5% reduction in toxicity when we're adding these microbes that we've identified. So these are really exciting results. What's even more exciting is that this was happening in a week. And as I showed you earlier, a lot of the previous work looking at OSPW detoxification has taken place over time periods of a month, 3 months, even a year. And so the fact that we're getting this initial large reduction in toxicity is really exciting to us. So next we wanted to scale up these treatments and take them from 200 ml shake flasks to that are being run in an incubator conditions to 60-liter fish tanks. And this allows us to perform fish toxicity testing following our microbial treatment. And so this is a picture of the setup where we have a control and then a microbial treatment. And you can see this turbid OSPW water. Here on the right, it's being aerated during an experimental trial. And really here we want to, when we're thinking about taking this to in the environment, it's great that our microtox test, which is working, but we want to know if we release the water downstream, is this going to be impacting the wildlife? And so here, fish toxicity testing is really the gold standard. Here again, we got really exciting results. So just to walk you through this graph for a minute, on the y-axis, we have a reduction in toxicity, and reduced toxicity, our target is this purple line where we have happy fish. And the way this fish toxicity testing works is that you put in the dilution of water required to reach a non-lethal concentration. And so with our untreated water, we need a 4 times dilution to reach this target. Whereas after our treatment, we need only a 1.3 times dilution. So the fish are almost happy, and we're working to make them happy. And like I said, this is really the regulatory gold standard, and it's a proxy for how the water would actually impact the wildlife, such as rainbow trout, during a release. Here again, we're continuing to work to optimize the treatment to achieve complete detoxification, because a complete solution is needed before this water can be released. At the same time, we are working to explore deployment options and to make plans for larger field scale demonstrations where we will likely work with partner companies to look at how to move these microbes really from the lab into the field setting. So in summary, we have discovered microbes that rapidly detoxify OSPW as measured by both microtox and fish toxicity. So far, we've scaled from 200 ml flasks to 50-liter tanks, and we have plans to scale further to start to think about deployment. And the microbial growth and corresponding toxicity reduction is fast. Our entire process right now is taking 7 days. We're iterating on this to work to completely eliminate the toxicity, but we're really excited about the starting point that we have. And OSPW detoxification has been a growing challenge for decades. In just 2 years, Allonnia has made really significant progress towards a solution. At Allonnia, we're really dedicated to using transformative biology to solve the world's toughest environmental challenges. We're always looking for new ones, so we would love for you to bring us yours. Thanks for listening today, and I would be happy to answer any questions at this point.
Awesome. What a fantastic presentation. Thank you for that, Michaeline. It really got my wheels rolling. So if you, like she said, if you have any questions, please go ahead and enter them into the software, and Dora Taggart and I will be sharing them for Michaeline to answer. So I actually already have one, so I'll go ahead and get started. So how do you think that, the question is, so how do you think that the open environment in the OSPW affects the microbial community? So like the sunlight, they mentioned the oxygen and different conditions that we'd see in this open environment.
Yeah, so I think that there are definitely a ton of conditions that are going to affect the OSPW and detoxification. And we've seen that in our experimental systems where we have the ability to manipulate kind of all of these different parameters, and we can, aerate a system, we can add nutrients, we can change the temperature, we can do it in the dark or in the sunlight. So what we're really doing in a lot of these experiments is figuring out the conditions where we can get the detoxification, and starting with conditions that work in the lab, and then slowly the idea is to then see, okay, if you move it towards more realistic field conditions, what are the key factors that we maybe need to think about for the deployment. For example, do we need to create a system or a deployment where we are having a system that's being aerated? Do we need to think about things like adding nutrients, or think about if we can alter the temperature? And obviously some of these controls are more difficult and more challenging than others, but that's definitely something that we're always thinking about as we're going about with our lab experiments.
Awesome, thanks. So for the next one, so I'm just going to read this all just so you have all the background on it. So this is how it comes in. From my understanding, the OSPW was filtered as stated. Was filtered OSPW spiked with salt to maintain elevated salinity TDS representative of OSPW in the field? Ask another way, is the halo tolerance of these degraders understood at this time?
Okay, yeah, so we have not specifically looked at the halo tolerance. I will say that, yes, a lot of our initial experiments, we did filter the OSPW. However, we moved from those conditions to not filtering it. And so, for example, once we were kind of moving up to the fish tank scale, which I showed you, that is all unfiltered OSPW straight from the pond. And so so the microbes that we're using are halo tolerant to the extent that they're able to translate from the filtered to the unfiltered OSPW.
Awesome. Okay, so here's another one. So what effect did adding carrier or biofilm bases have on detoxification?
Yeah, so that was something that we started out looking at, and honestly, it's not one of the solutions that we ended up going with. It was one of our initial kind of test conditions as we were doing the enrichments, and there's previous literature kind of supporting that. For us, it wasn't something that we ended up continuing down the path with, because we found other isolates and consortia that were not biofilm based that seemed to be performing better. But it is an interesting idea and something that could be explored further.
Okay, so so here's another one. So are there other ways besides water, such as sludge, and would you comment on treatment of such media?
Sure. So I guess as you get into something like sludge, you'd have to think about altering. It goes a little bit more into the deployment and how you might deploy a solution, and also to the environmental conditions. So is the sludge kind of a consistency that could be aerated? Is it going to be aerobic or anaerobic? There's also potentially there's more potential for the naphthenic acids to maybe stick to the particles. So there's a lot of questions around those lines. So I think that if we were to explore sludge, or kind of translating from more of the water setting to a sludge setting, you'd have to think about some of those differences between the water and the sludge to see if the microbial solution that we've developed is still working.
Okay. So here's another. So is the bacterial culture that you have developed an aerobic culture, or is it relying on anaerobic processes? And what are the electron acceptors needed, and how would you get the appropriate quantities introduced?
Yeah, so right now we're working in aerobic systems. So there is a diverse community in OSPW. We're really focusing right now in targeting systems that are aerated.
Okay, awesome. So you mentioned this treatment is very fast, 7 days. So are you finding that this is consistent across water impoundments and treatment zones, and where it is being used, or are there other factors that come in to the time? And do you feel like, could this have like a, like with environmental remediation, a lot of times, we see a lag or rebound? Do you expect something like that?
Yeah, so we have a lot of our initial work was done on kind of a specific set of water from one oil sands company provider. We have gotten water from another location. So we're starting to test water from different locations, and that's definitely an area of active research, because we do want to know the answer to that question: is how specific is this going to be to a specific water composition? And at this point, I don't have the answer to that, but stay tuned, because that's something that we are kind of working on actively right now.
Great. So in that case, so do you feel like this could come to something where you're going through like a series of treatments? Is that kind of the vision with this, or combining different things? Like I know within the water, a lot of times they're using electrolysis to introduce oxygen. Have you guys
Mm-hmm.
looked at any kind of combination treatment or something like that?
Yeah, so that's definitely something that we're thinking about, is that there's kind of, through the detoxification is likely, it's a big challenge. There's going to be different steps of what's needed to make the water tenable for release. And so, yeah, we're definitely thinking about how this might be combined with other technologies for complete detoxification and for release.
Awesome. Well, so I think so let's see, one more. So was naphthenic acid concentration measured through 7 days with any different analytical techniques?
Yeah, so we have done some measurements of naphthenic acids, and we see some decrease with our microbial treatment. We also, there's still naphthenic acids present, and so I guess that's something else that we are currently exploring, is that it seems that some of the toxicity reduction that we're seeing is associated with a naphthenic acid reduction. However, our hypothesis is that there is other toxicity reduction that's occurring that is potentially associated either with naphthenic acids that we aren't measuring right now with kind of the classical measurement techniques, or that there's other toxic organic compounds that are also contributing to that toxicity that our microbes are targeting. So it's both. We're seeing that they're targeting some of them, and we're trying to figure out what else the microbes are targeting in the water.
Great. All right, so do you know of any biological processes that produce thiophenol in these types of settings?
I am not, no, I'm not aware. It's not really, I guess I haven't thought about that, and it's not an area that I know a lot about, but I would be interested to learn more.
Awesome. Always something you can get back with later or something if somebody would like to contact her directly.
Definitely.
Consists of a treatment plan. All right, so I think we're getting to the one more. Are there energy inputs for heat, warmth, and aeration? Kind of like what I asked earlier. So if you looked at any of that kind of thing.
Yeah, so I mean, right now we're really thinking that, for example, we might not want to do something with heat because that's going to be pretty energy intensive. And so we're focused on trying to keep things at ambient temperatures or even cool things off. In terms of aeration, there's a lot of different ways to think about that. I mean, one way is just thinking of a flow gravity system where you can kind of aerate the water. Another thing is wetlands we've thought about in terms of a deployment scenario where you can have aeration through the plant community. So we are thinking, especially in terms of the question of aeration, we are thinking about how we might be able to make the system, to aerate the system in ways that are kind of lower energy to allow for increased microbial growth.
Okay, so did you sequence the enriched microbial consortia, and what's the community difference?
Sure, yeah. So we have done some sequencing on the enriched consortia, and we generally see that the community decreases in diversity to some extent, and also the composition shifts, and there are certain microbes that start to do better and to grow better in our enrichments. And I guess that's about as much as I can tell you on that topic.
Well, that's great. Did you guys start with a pilot scale study, and were you trying to, beef up or feed, the wild native population of microbes, or were you looking at engineered more so?
Yeah, so we have not gone into a pilot scale study. Right now, that's the next step, is to try to take it into a mesocosm and field scale, and then field scale study, hopefully in the next 6 months. However, right now we're really focused not on engineering the organisms, but on more, or at least not engineering in a lab, more doing kind of adaptive evolution, trying to get the organisms to perform better in environments we're interested in, partially because of regulatory issues, and really wanting to get these solutions out into the field in the near term. Awesome. And that's a lot easier when you're thinking about using what's already there in the natural community and working from that.
Okay, so another one, do the bacteria introduced have an impact on the overall environment? And like, in terms of, do you feel like there's a potential for them to spread outside the area that you treat? So is there any concern with that?
I think right now that's not so much of a concern, especially since we're really focused on microbes that, again, are already in the environment and are kind of natural inhabitants of these OSPW systems. It's obviously something that we could think more about, but the condition in the future, if we were to kind of go down that engineering road, but as of now, using these natural communities and really just trying to augment certain members, we're not as worried about that, because these members are kind of going to grow well in the augmented conditions, but are unlikely to continue to expand once they're outside of those conditions for their optimal growth.
All right, well, I think that's it for today, Michaeline. It's a really great presentation. It's fascinating. Felt like I learned a ton. I'm sure everyone else feels the same way.
Great. Well, thank you very much for having us, having Allonnia present, and yeah, please feel free to also email any follow-up questions.
And again, thank you to everyone who was able to join us today. And please look for a follow-up email in the next few days with a link to the recording, as well as a link for a certificate of attendance. Until next time, stay safe and awesome, everyone. Thanks.